right, thank you very much for the nice introduction and for having me here for one of these PerfLab seminars in the NHR. So as you said in the introduction, I have actually two appointments. I'm jointly appointed by the TU Dresden, the Dresden University of Technology, and by the Max Planck Society. I was appointed as one of the directors of the Center for Systems Biology Dresden, which was newly founded in 2012. And in 2012, I moved to Dresden as one of the two founding members of this center. Before that, I was a professor in the Department of Computer Science at ETH Zurich. And also now my professorship at the TU Dresden is in the Faculty of Computer Science. And I'm also currently serving as the Dean of the Faculty of Computer Science. So don't be afraid by the name Cell Biology and Genetics. I'm actually a computer scientist, and I'm going to talk about data structures and things related to high performance computing, but of course with applications in biology. And I want to start out by showing you this beautiful picture of the building where I'm sitting in now. This is the Center for Systems Biology Dresden. So I'm actually sitting right here. This is my office here, which is dark here on the picture, but it's because the sun is shining outside. And I'm talking to you from there. I would have loved very much to travel to Erlangen, but my schedule did not allow, but maybe we find another occasion where we can make this happen. In my group, we study emergent behavior in dynamical systems across scales. And of course, these are, again, living systems or biological systems. One example is emergent dynamics in active particles. So what you see here is a microscopy image of fluorescently labeled motor proteins. So these are individual single molecules that can walk on polymer filaments, and they consume or they hydrolyze a chemical fuel, and then they can turn that into mechanical stresses and forces. And one thing that happens is that it forms these nice spontaneous density bands here. And that's an interesting emergent phenomenon because nobody is telling these proteins that they have to form bands, and one would actually expect them to maybe disperse uniformly. And so this is an experimental image, a microscopy image taken in the lab of Erwin Frey at LMU in Munich. The same can occur also on the cellular scale. This is a microscopy video from the lab of Stefan Grill here in our Max Planck Institute, showing two proteins in two colors, one in red and one in cyan, that initially are uniformly distributed in the cell, but then spontaneously segregate to the two poles of the cell and stay there. And actually the cell divides along the midline so that one daughter cell will inherit the red proteins and one will inherit the blue proteins. And this sets up the head-tail polarity of the embryo. Here is another example from the lab of Pavel Tomanchak. This shows a Drosophila, a fruit fly embryo during development, and it's a large three-dimensional microscopy video showing the development of the tissue. And the tissue flows and the tissue folds and forms the inner organs and flows around the back of the fly. And again, we can ask the question, how do the cells know in which direction to travel? How does a cell know when to divide? How does a cell know when to differentiate and in what way to differentiate, such that this does not become an amorphous ball of cells, but a well-formed embryo? And of course, you can ask the same questions on the ecosystem scales, be it microbial ecosystems like microbiota, or also terrestrial ecosystems. This is from the lab of Knut Drescher at the Max Planck Institute for Terrestrial Microbiology. How do different species interact with each other, and how do spatial patterns emerge and maintain in ecosystems? And in all of these scales, on all of these length scales, we abstract the individual constituent as what we call a particle. So in an ecosystem model, a particle would be an individual animal or an individual bacterium. In the cellular level, a particle would be an individual protein. Here, a particle would be an individual molecule. On the tissue scale, maybe a particle could be an individual cell. And then we ask the question, how do these particles need to interact with each other in order to give rise to the observed emergent dynamics on the macroscopic scale? So the velocity fields that we see, the deformation fields that we see, the concentration distributions that we see. And there are two types of questions that we can ask. We can ask what is a minimal but sufficient set of interactions that are sufficient to describe the emergent dynamics, or we can also ask which interactions are necessary, meaning without which the dynamics could never emerge or could never be described. And of course, you do this by combining theory and experiment. And some of the key questions are, what are the physics of the interactions? Like, are these interactions mechanical? Are they chemical? If they are mechanical, what sort of mechanics? Is it stress sensing? Is it hydrodynamic coupling? We can ask the question, what is the correct description of the emergent dynamics? We see some velocity fields and we see some concentration fields. There are also fields that we don't see. We don't see, for example, in the microscopy video, the force field or the pressure field, which is obviously there. And we can come up with different models, with different physical models for those. And of course, ultimately, we want to be able to predict or explain the emergence of how the interactions actually give rise to these emergent dynamics and exactly which interaction is responsible for which aspect of it. And as I said before, we do that by combining theory and experiment. We have experimental data, and these in our case are microscopy videos, three-dimensional microscopy videos. And the theory we have on the other side in our case is most of the time partial differential equations. And then in my group, we do research in algorithms that can be used to infer theory from data. So algorithms like symbolic regression algorithms for learning equations out of videos, or physics-informed neural networks and stencil nets for learning numerical approximations out of data. And we use numerical solvers or simulation algorithms to numerically solve the theory equations to predict what kind of dynamics or data they will generate. And then this is put in a classical reverse engineering loop. So we have a microscope, for example, here in the building. We have a lot of so-called light sheet microscopes, which produce three-dimensional, very high resolution, three-dimensional images of developing tissues and embryos. And they do that over the entire time span of development at sub-cellular resolution. For example, here you see a fish embryo. Fish are very nice model organisms because they develop in these little fish eggs that you may know. And these fish eggs are transparent, so you can put them in the microscope and the fish embryo develops inside the egg, and you can image the embryo over its entire development without disturbing it. And also fish live in water, and these microscope chambers are usually filled with water to have the correct optical properties and refraction index. And so what the first thing is that we do is we visualize these microscopy images in 3D, because these microscopes acquire three-dimensional volumes. We want to also visualize these as three-dimensional volumes, and we use virtual reality and augmented reality techniques for that. This picture here is from a newspaper, and this newspaper has taken this photograph in the basement of our building here of the CSPD, where we have a four-sided virtual reality cave where the experimenter can immerse him or herself into these 3D images in real time. So the images are displayed while the microscope is acquiring them with a very low latency. And that requires very high-speed real-time rendering. Our virtual reality cave, if you sum up the pixels on all the four sides, and you need 120 hertz, 60 for the left eye and 60 for the right eye, you need almost 1.8 gigapixels per second real-time rendering performance. These things can also not be pre-rendered because they depend on the viewpoint of the user. The second thing we do is we can take these images and we can feed them into a high-performance computing system, which we also have one here in the CSPD and an even bigger one at the TU Dresden. And these microscopes produce images at currently more like 5 gigabytes per second. So they're usually between 3 and 4 cameras attached to a single microscope that are taking angles from different sides. All of these cameras have very high resolution, and all of these cameras produce three-dimensional depth images, and these images can be then fed into a computer. And one thing we can do with that, and that we have done already almost 10 years ago, is we can run computer vision algorithms then to segment, for example, these images in real time. For example, in this example here, we could segment the vasculature of the fish, which you see here highlighted, the blood vasculature. And then we can also run in these segmented 3D geometries numerical simulations, for example, of blood flow. And ideally, the result of that simulation would be superimposed onto the data in the virtual reality display with a latency that is below 1 second. So the user can actually see how the blood cells are moving through the vasculature and can also see the velocity field from the simulation. And the simulation can then be used to infer fields that we cannot see, such as, for example, the wall shear stress or the pressure. And for this, we develop software, we develop Scenery as a big open source project for virtual reality and augmented reality computer graphics. We develop PPMRC as a real-time computer vision platform, and we develop the OpenFPM framework for highly scalable simulations, numerical simulations. And that works then in a loop. Yeah, we start with a real-time observation of a three-dimensional microscopy video. We have a computer simulation of a biophysical model. We can display them in an VR environment for the user to compare. The user can then give feedback, or we can also compute other loss functions that can be fed into a machine learning algorithm. And this machine learning algorithm, again, tunes the parameters of the computer simulation. One thing I'm not going to talk about today, but that we have done last year, which is, I find quite exciting, is also this red arrow here, that we can directly from the video use machine learning to get a simulation model without first deriving an equation and then fitting the parameters. So you directly fit the equation. And all of that is based on a high-performance computing platform, which in our case is OpenFPM, the open framework for particles and meshes, which we have been developing in the group for almost 25 years now. It previously was called the PPM library, the Parallel Particle Mesh Library. And about 10 years ago, we have renamed it to OpenFPM, and we have re-implemented it in C++, making heavy use of template metaprogramming and compile time code generation to target the library to different hardware platforms, from graphics cards to distributed and shared memory processors and vector processors and so on. And this is still ongoing research, and I will talk about some of that later on. We have also put on top of the library a domain-specific language together with my colleague here, Jeronimo Castrillon, who is the professor for compiler construction, which, for example, provides a nice way of implementing partial differential equation solvers in a nearly mathematical notation. But the big problem that we had to face first was, how do we deal with these very large images? How do we even visualize them? How do we transfer them? How do we compute on them? And so what you have to imagine is what I try to show you here with these little circles. These are maybe different cells in an embryo. And then these microscopes acquire image planes. So you acquire one plane, and that plane is then stored as a two-dimensional image, which is an array of pixels. And on the right hand, you see what such a plane can look like. This is a plane from the lab of Jan Heusken, a light sheet microscopy plane where you illuminate with a planar light sheet, and then you image perpendicular to it. So you really get one section through the embryo. Again, this is a fish embryo, and you see the cells here at fixed depth. Now we do this for multiple planes. The illuminating light sheet moves across the sample. So we acquire multiple planes and we stack them together into what is called a Z-stack, or a three-dimensional image. Okay, and this three-dimensional image of the same fish embryo then looks like this. The color here simply codes depth. So things that are more blue are further away from the camera position, and things that are more yellow are closer to the camera position. And then you can do this over time. You can require one such Z-stack every second or every few seconds, and put that together into a movie, which shows the development of of the embryo. So this is again a fish egg. You have a cell mass growing on one side, the cell mass then develops around, cells differentiate, you start to form here in bright colors the spine, the spinal nerves, the spinal cord, the tail of the fish develops, the head of the fish develops, the inner organs start to develop, and you can really appreciate the development of a complete organism over its entire lifespan. And so these are, of course, fantastic data sets for biology. And the current challenge is to track all the cells, segment all the cells, and learn something about how cells take decisions and how they coordinate with each other when they form such an embryo. But first of all, you have to deal with the data size. Each of these 3D images is about 4 gigabytes, so about 10 to the 9 pixels. So these are, a few hundred planes. Each of these planes is 2048 by 2048 pixels. And then you do this also over time. So the video that I have just shown you here has a size of 2 terabytes, roughly 10 to the 12 pixels in this video. And of course, the sampling rate that we use for this, the size of the pixels, the magnification that we set on the microscope and on the camera has to be tuned such that it resolves the smallest detail that we are interested in. And here, the detail we are interested in are the individual cells, the individual nuclei. So we would use a pixel size that is maybe not bigger than the blue bar here. And because we do this on pixels, and pixels have the same size and the same shape everywhere in the image, we get a lot of pixels, and all these pixels have the same size and the same shape, and they all are small enough to reduce the smallest detail in the image. And of course, this gives x times y times z, a lot of pixels, times t, even more pixels. So this is where these huge data sizes come from, and this 2 terabyte video is actually already from several years ago. We have projects now where the videos that are acquired going to the petabyte range. So we need a computer to get the information for us, but even visualizing such a large video is a challenge because you cannot simply load it into the texture memory of a graphics card and then use standard computer graphics algorithms. You need to do something smarter to even get this rendered or visualized. And if you want to do computer vision on that, it becomes even more tricky because, again, not only are all the pixels of the same size and sometimes maybe unnecessarily small, but they also are all of the same shape. And they're only distinguished by the number that they store, which is the local intensity value. So the image really looks like this big table of numbers, and we need to ask, what is information and where is the information, and can you squint your eyes hard enough to see that there is actually a nucleus here? Right? And so this is why so many computer vision tasks are hard, because pixels are really maximally uninformative about the definition of information that we have in the image. Okay, so getting at the information is hard if you have data like this, which is somehow stored on a uniform data structure and which is large. And so this is where the idea came of storing the images in a more adaptive way. And this is the adaptive particle representation, where instead of using pixels that are the same size and the same shape everywhere, we only place collocation points in the image plane where they are needed. And we call these collocation points particles, in analogy to particle methods in numerics. And so, for example, this same image here could be represented like this. We replace particles very sparsely in the background, which is mostly black, and we place particles more densely, and maybe smaller particles, if we can define something like a particle size, in areas where there is stuff in the image. And the question, of course, is, given an image on pixels, how do we determine the sizes and locations of the particles that are required to represent the same image? Ideally in such a way that the overall number of particles that we need is as small as possible. And this was the PhD thesis of Bevan Cheeseman in my group, who came up with this idea of the adaptive particle representation. And we wanted the adaptive particle representation to satisfy 4 design criteria. We wanted it, of course, to adapt to the content of the image in a way that controls the error. So we wanted to be able to set a worst case bound on the reconstruction error and be mathematically guaranteed that that is always satisfied point-wise everywhere in the image. We wanted to take into account the local intensity scale in the image. So to represent dim and bright objects with the same importance. We want it to be fast. Fast in this case means linear in the number of input pixels. And we wanted to support direct processing. We want that you can directly do all the downstream image analysis and image processing and rendering and visualization on the APR without ever having to go back to pixels. So the first question, of course, is, did it already exist? What we came across in the literature in image-based systems biology are mainly 2 approaches: spatial projections, which is a rather simple approach where, for example, this fish embryo, you say, okay, the fish embryo initially is like the surface of a sphere. So instead of storing the image in 3D, I store it in spherical coordinates and project it onto a sphere and then only store it in 2D. Now, this, of course, reduces the amount of data by a great deal, but it is only applicable to simple shapes, simple embryos that can be projected to a lower dimensional manifold. And the second approach is that of supervoxels or superpixels, where you take neighboring pixels, you look at their intensity, and if the intensity is similar in neighboring pixels, then you group them into one bigger pixel. And you do that, and you build a tree where you have very large pixels if things are very similar, and very small pixels where things are not so similar. In other fields than image-based systems biology, of course, people use wavelets, they use multi-resolution pyramids, they use in numerics equidistribution principles and adaptive Lagrangian particles to have spatially adaptive discretizations, for example, in PDE solvers. The adaptive particle representation is different from all of these. It's of course different from spatial projections because it actually is 3D and it does not require reducibility to a lower dimensional manifold. It's different from supervoxels because supervoxels are constructed in a greedy way, whereas the adaptive particle representation is globally optimal. It's different from wavelets because wavelets do not allow direct processing. Wavelets are a frequency-space representation of the image. And if you, for example, want to render the image on screen, you need to transform it back. And in the APR, we never transform back. It's also different from equidistribution principles and Lagrangian particle methods because, again, the Lagrangian particle methods are not globally optimal. And it's actually most similar to equidistribution principles. And so here is how it works. This is a very simple example where we have 2 objects that are bright and a background that is dark. If we take a cross-section through this 2D image and plot it in 1D, because it's easier to draw on the slide, then this is what the intensity signal as a function of the line coordinate along the dashed line would look like. The highest detail we have, of course, near the edge of an object. There we want to have a small pixel size, so we actually accurately represent the shape and the location of the edge. That corresponds to high information. We have relatively medium information in the interior of the objects. There may be some shading, some variation of the intensity inside the object, which may tell us something about what is going on in here. And we have the lowest information, of course, in the background, which is ideally just flat and black. So we want to adapt the pixel size to the amount of information that we have locally and use large pixels in the background, use small pixels here, and maybe medium-sized pixels inside. And we call these pixels particles. So a particle, again, is a generalization of pixel that can have any size and any location. Or for those of you who are familiar with basic numerical methods, it's a collocation point. And so if we zoom into this tip of the signal, then we have here the fluorescent signal. Let's say we want to represent the signal at the blue point, and then we could use a particle with a certain size. And if we do so, we represent the signal here by a particle, and we do the simplest possible signal representation, which is piecewise constant. So we do a 0th order interpolation. So we say inside the particle, the intensity is just uniform and equal to what it is at the sampling point. This is the simplest and least accurate interpolation scheme. Then we get the certain error, and the maximum error we get here. The maximum error is the largest deviation between the actual signal and our representation. Of course, if you make the particles bigger, then this maximum error grows. And if we make the particles smaller, then this maximum error shrinks. And in general, the magnitude of the error, at least to linear order, depends on the gradient of the signal at the collocation point. If the gradient is steeper, then this error will grow faster as we move out and vice versa. And if we make the particle smaller, the error becomes smaller. And so what we decided in the APR is a very simple criterion. We let the user define an error threshold, and we call that E star. And E star should be the worst case reconstruction error. So if we take the APR of the image and interpolate using the least accurate interpolation scheme, which is piecewise constant, the intensity anywhere in that image, the deviation from the original pixel value at that location should be less than E star. And we can discuss maybe afterwards what that means and whether this is appropriate. But in that sense, the APR is a representation of the same continuous signal as was represented on pixels before, but it's not identical, of course, to the pixel image. But even the pixel image is, of course, only a discretization of the actual continuous image. So there is no ground truth here. And in practice, we set E star to be smaller than the noise level of the camera. So everything we lose is noise. And in that case, the APR also has a nice property, which is denoising, implicitly denoising the signal. So anyway, so we say that at this location, we want to deviate at most E star, which is these two dashed lines. And then we want to find the largest particle size such that the error everywhere within the particle is below E star. And so that would be the largest particle size. If we make the particle this size, then we have an error of E star exactly at this point. Everywhere else, the error is below E star. And if we would make the particle just a little bit bigger, then the maximum error would exceed the threshold. So that's how the APR works. The APR basically finds the locations and sizes of particles everywhere in the domain, such that at every point, the maximum error for the most stupid possible interpolation is guaranteed below E star. But this has a problem. If one object is brighter than the other, this would lead to, and we have the same E star everywhere, this would lead to dim objects being represented with less spatial resolution than bright objects. Because we can be within E star in a dim object with a larger particle than in a bright object, but we would need a smaller particle. So implicitly, if we just set the error threshold to be the same everywhere, we would start to under-resolve dim objects and over-resolve bright objects. And we don't want that. We want that every cell and every piece of signal in the image is represented with equal importance. So the idea here was to define something like a local information scale, which is defined by the point spread function of the microscope, but it's just a local signal to noise ratio in the signal. So it's basically the amplitude of the signal. So how much brighter is the foreground than the background locally. And this local information scale is used as a measure for the error threshold. Okay, so we bound the error relative to the local information scale. So instead of bounding the absolute error of the reconstruction, we bound the relative error of the reconstruction, and it's relative to the local information scale. And the local information scale, of course, is bigger for bright objects and smaller for dim objects. So if you multiply E star with that, then the size of the pixels that you get out for both is, again, exactly the same. And so by doing the error threshold with respect to O, we can get the same spatial accuracy for objects of different brightness. Okay, so we have these two ingredients. We have the gradient of the signal, which we need to compute, and we have the local information scale, which we need to compute. Of course, the signal in a real image can be noisy, and computing gradients and information scales may be dangerous, because if you just take a finite difference, for example, you will amplify the noise. So in order to do that in a way that is less sensitive to the noise, we use smoothing B-splines. So we represent the whole image first by a smoothing B-spline, and we then use this smoothing B-spline to compute the gradient and the local information scale. But then we go back to the original image and transform that onto an APR. So we need these ingredients. We need a user-defined error threshold, which is pointwise. We need the gradient of the image, and we need the local intensity scale. The green one is given by the user. The two white ones are computed using filters on the input image, and that gives us the size h of the particles and the location x of the particles everywhere in the image. And Bevan first had a rather frustrating experience because he analyzed this problem mathematically, and he found out that this is not efficiently solvable. This is actually NP hard. It's NP hard to find the location of a set of particles and the sizes of the particles such that overall you need the smallest number of particles to satisfy a given E star. But Bevan then found out that under two restrictions, the first one being that particles can only change size by factors of 2. So we have a dyadic binning. So the second largest particle is 2 times the smallest, and then times 2 times 2 times 2. And the second condition is that neighboring particles can only differ at most by 1 resolution level. So you cannot have a big guy neighboring a very small guy, but they can only differ by 1 resolution level when they are next to each other. And under these two conditions, that particle sizes are powers of 2, and that neighbors differ by at most 1 resolution level, Bevan could actually mathematically prove that the problem can be globally optimally solved in linear time. So the APR algorithm, which is called the pooling scheme, takes these ingredients and computes the particle sizes and positions under this constraint in a way that is globally optimal. So there is no other dyadic representation of the same signal that would need less particles or less points to achieve the same accuracy. And that's nice. And because it's still a real space and not the frequency space representation, we can directly use this adaptive particle representation of the image now to do downstream processing. And so this is what we call the APR, or adaptive particle representation, in 1D. And of course, you can do it in 2D, you can do it in 3D. In 2D, the particles are then the particles are more like rectangles, and in 3D, they are more like boxes. The particle is the point at the center, and then the size of the particle is basically defined by the size of the volume of space that it is responsible for representing. Okay, the practical algorithm works like this. It takes any input image on pixels, which is an intensity as a function of space, and a user-defined error threshold. It then computes the local intensity scale using these smoothing B-spline filters, and it computes the image gradient using the same smoothing B-spline filters. It then combines the two to give a pointwise estimate of what resolution H would be required everywhere. And then it uses the pooling scheme to start from the lowest resolution and build the tree up to enforce the two conditions that particles have to be powers of 2 and neighbors can only differ by 1 level of resolution to give you then this multi-resolution tree and then represent the image on that. What you see here on the right is on the left part, you see the actual particles color-coded by intensity, and on the right-hand side, you see the actual image represented by this. And these particles carry more information than just the raw image, because what I do here, for example, is I just plot the particles as blue dots on a certain resolution level. And you see that this basically already segments the individual cells in the embryo. So the hope is that also computer vision tasks like image segmentation become easier because the particles are already located to where the objects are. So does it fulfill the representation criteria that we had? We tested this experimentally using synthetic 3D images and also using 19 exemplars of real-world microscopy data sets. The first reconstruction condition says that the error in its infinity norm has to be pointwise less than the user-defined threshold. So we tested that across all data sets using the least accurate interpolation, which is piecewise constant. Again, particles are just flat. And we saw that the actually observed relative error at each point is indeed less than the user-defined error bound for all the data sets. The next is the adaptation with image content and image size. So for that, what we did first is we fixed the image size and we increased the information in the image by making synthetic images that contain more and more objects. So we just put circles and spheres into a synthetic image. The image always was of the same size, so always 1024 by 1024 pixels, but then we put more and more objects into this image, and we see that the number of particles that the APR uses grows proportionally to the number of objects that is actually in the image. The second test is you keep the number of objects in the image constant, but you increase the image size. You resolve the objects with pixels that are smaller and smaller, so you use more and more pixels, but it's always the same object. And what you also see here is that as soon as the object is well resolved, is fully resolved, the APR needs a constant number of particles because it just represents the object. And if you oversample the object with too many pixels, it throws them away. Whereas the number of pixels would, of course, grow with the cube of the image width. So RC1 and RC2 are fulfilled. RC3 says the APR must be computed in linear time. Easy to test. We take images with different numbers of pixels, convert them to APR, measure the time. It's indeed linear. Interestingly, less than 5% of the time is actually spent on the APR algorithm, and 85% is spent on the B-spline filters in the beginning to compute the gradient field. And the remaining 10% is spent on reading the data from the file. Across the 19 real-world data sets, we achieved the conversion time that is 3.6 seconds on average and maximum 7 seconds. So that's good because 7 seconds is actually faster than the microscope can acquire such a volume. So we can use this in real-time settings and we can directly transform these things at the microscope before the microscope comes with the next volume. And it compresses these things a great deal. So instead of, for this zebrafish video here, instead of having 2 to the 9 pixels, we now have 10 to the 9 pixels. We now have 10 to the 7 particles. So instead of 4 gigabyte of data, we have 49 megabyte. And this is the head of a fish embryo. Again, across all 19 exemplars we tested, the mean data size went down from about 1.8 gigabyte to about 49 megabyte. And we on average needed 40 times less particles than pixels. So typical compression ratios, the ratio between the number of particles and the number of pixels for microscopy images are in the range of 10 to 100, between 10 and 100. And of course, the APR can directly be visualized. This is a direct raycast, a simple raycasting algorithm through the original pixel volume and through the APR volume, and they look identical. But the lower one can actually be computed fast enough so you can use it for virtual reality. And finally, can we use the APR for processing tasks? We argue yes. The APR does not only reduce the memory, but it also reduces the computational cost, because all downstream processing like neighbor access, filtering, segmentation, visualization only needs to loop through the particles, and there are less particles than there were pixels before. But of course, the data structure in which these particles are stored is more complicated. But it's possible. We can use the APR directly for downstream processing. And I argue that the downstream processing tasks sometimes become easier because the particles do not only store an intensity, but they also have a size and a location, which, as I showed you before, already kind of shows us where the objects are in the image. They also have additional information, like particles know which resolution level they live on. Particles know what resolution level the neighboring particles live on. So there's a lot more information about where things are in the image, which makes the computational problems also sometimes algorithmically easier to solve. And if we loop through the particles instead of the pixels, we focus the computation on those parts of the image that are actually important, that are actually informative. Recently, we then used this to define mathematically consistent multi-level discrete convolutions on APRs. So we can take an APR of an image. The APR, of course, as you see by now, can be represented as a tree because particles live in these dyadic bins. And neighbors only differ by at most one level of resolution. So you can write this, you can interpret this as a tree. And the question then was, how do we compute a convolution on the different levels of the tree such that the result we get is the same as if we would do the convolution on the full pixel image and then only do the APR. So this is what we call as a consistent convolution, and that's in this transactions paper here. And what we used for that was earlier work from our group, which we presented at ISC about sparse block grids on GPUs, where each level of an APR is represented as a sparse block grid. And these sparse block grids are stored in a compressed sparse row matrix format. And then we can compute the convolutions on these things on graphics cards. Of course, we save memory. This is an interesting plot. So this is actual memory usage measurements from a benchmark, where we have a pixel image that is about 4 gigabytes. If we compute the convolution on that, we need 2 times that memory. We need the memory for the input image, and we need the memory for the output image. If we do it on an APR, which basically does not save us anything, the APR has as many particles as the original pixel image had pixels. Then we have an overhead because we need additional memory to store the APR tree data structure. And we need an additional overhead for, the tree data structure is the purple and the green. And we need an additional overhead for storing these fast access data structures, these sparse index data structures that we can have fast random access into the tree. But as soon as the compression ratio reaches 2 or 3, we are already more than a factor of 2 lower. And again, for typical compression ratios, which are on the order of 40 to 60, 70, the memory requirement is greatly reduced compared to doing the processing on the pixel image. And because the downstream processing is also accelerated, we can actually reach throughputs on the order of 1 terabyte per second on a single consumer GPU. So this is not an expensive GPU. This is a gaming GPU, an RTX 2080. And the blue and orange line here are the throughput that we get for different computational ratios on the APR, and the horizontal blue line and dashed line is what we get if we do it on pixels. The two lines are for convolution kernels of different size, so for a 3x3 kernel and for a 5x5 kernel. The red and green, we don't need to look at. This is if we do the thing on the CPU using all cores of a CPU with OpenMP. And so we can implement now any kind of image processing algorithm that is based on convolutions, for example, deconvolution, Richardson-Lucy deconvolution. This is a ground truth image of polymer fibers. This is the input image, which is blurred by the point spread function of the microscope. This is the Richardson-Lucy result on pixels, and this is the Richardson-Lucy result on the APR, without ever going back to pixels in between. And again, they're the same. If anything, I would say the APR is even slightly crisper because it has a denoising property. And in a recent collaboration with the Wyss Center for Neuroscience, we're applying this now for digital anatomy. The goal is to image an entire human brain at sub-cellular resolution. This is the 42 petabyte image that I mentioned initially. And here is just a comparison of how long it takes to do the different processing steps on pixels versus on the APR. So a single microscopy cube, which is a 1 cubic millimeter piece of brain tissue, is 2.4 gigabyte, and it goes down to 20 gigabyte on the APR. Stitching the cubes together, like finding which cube goes where and stitching this together into a whole brain, goes from 3 hours and 15 minutes to 55 seconds. Segmenting all the neurons using a machine learning algorithm goes from 80 hours to 1 hour and 20 minutes. And mapping these regions to a brain atlas goes from 15 hours to 4 minutes. So here the APR really enables a pipeline for very high throughput and very high resolution digital anatomy. And you can then look at, for example, amyloid beta plaques in Alzheimer patients. You can see individual plaques, you can see individual cells, and you can do all of this very fast. And it scales to a whole brain and potentially even to multiple brains. And finally, we have the scenery software, which can be used to visualize these things also in real time. On the screen, there is a Fiji plugin called SciView, but also on headsets and on room-sized installations. Because the code is distributed memory parallel, it can also run on multiple GPUs. In the cave, for example, where each projector is driven by its own GPU. And we have such a cave, and here is a bunch of school kids. This is my son. And they look at the Drosophila embryo here developing in the microscope, and they marvel at the tissue folding and at the decisions of the cells. So to conclude, large and sparse images can be represented using the adaptive particle representation. The APR is globally optimal and gives guarantees a user-provided point-wise reconstruction, worst case bound. It can be computed in linear time and compressed in memory. It supports native image processing, saving both memory and compute time, and it saves orders of magnitude in compute resources, both memory and computer time, for large biomedical imaging studies like the whole brain imaging. And of course, I also like to say that it saves a lot of power usage and CO2 because we only need the cluster for 2 hours to process the brain, not for 2 weeks. All our software is open source. We have the Mosaic suite for Fiji and ImageJ for all the image analysis parts, tracking, segmentation, enhancement, and so on. We have the OpenFPM platform for highly scalable simulations. We have the APR, which I talked about today, which comes in the form of a C++ library, a Python package, and also an APRs plugin. We have machine learning algorithms to learn models from data, and we have the virtual reality environment scenery. And with this, I would like to thank all the people in my group and all the collaborators, and of course the funding agencies. In particular, today I talked about work that was done by Bevan Cheeseman, who was a PhD student in my group and developed the APR originally, and Joel Johnson, who is a PhD student in my group right now and does the APR native processing and APR convolutions and APR visualization. And the brain imaging project was done in collaboration with the Wyss Neuroscience in Geneva and the University of Geneva. And this is our website where you can find the code, the papers, and everything else, and our Twitter or X handle for announcements. Thank you very much.
So thank you very much for this very inspiring presentation and talk. This is really interesting work, and which underlines how important the choice of good algorithms is in the context of high performance computing in the context of large scale image processing.